Quorum sensing in bacteria in plain English. Many people think that bacteria are simple, primitive organisms that live an asocial life due to the fact that they are single-celled microscopic organisms. This of course is incorrect. Although these microscopic organisms are often single-celled, in groups they are capable of carrying out an array of processes from bioluminescence to pathogenic attacks. How is it that these seemingly simple organisms can do such amazing things? The answer, simply put, is by communication. Bacteria are able to communicate with one another through a process known as quorum sensing. Quorum sensing is a phenomenon in which bacteria produce, release, and detect chemical signals. The chemical signals are known as autoinducers, which here are represented by the black dots, and are similar to hormones or pheromones. Once produced, they are released to the outside of the cell, and other bacteria are able to detect them. When a bacteria is in a low cell density population, it still secretes autoinducers, but at a lower rate. However, when several bacteria are in the same population, this is known as a high cell density population. As the bacteria detect the rise in population density via autoinducers, they in turn secrete more autoinducers proportionally. When the amount of autoinducers in the immediate proximity reach a certain concentration, they pass a threshold and the cell's receptors become fully activated. Activation then induces regulation of certain genes, such as the bacteria in Vibrio fischeri, which produces bioluminescence in the squid via quorum sensing. A major division in bacteria is the gram-positive and the gram-negative bacteria based on the presence or absence of a cell envelope. The class of bacteria determines the autoinducer that it produces and secretes, and also the signal components within the cell. Gram-positive bacteria sense and secrete oligopeptides and have a two-component signal transduction system. In general, the peptide is secreted by an ATP binding cassette transporter that is found in the outer membrane. Detection of autoinducers is controlled by two-component adaptive response proteins known as sensor kinases. The interaction of peptides with the sensor kinases causes a series of phosphorylation which results in the phosphorylation of a response regulator protein. This activates the regulator, allowing it to bind to DNA and alter transcription. Gram-negative bacteria sense acyl homoserine lactone with Lux-I and Lux-R complexes. In general, the acyl homoserine lactone signaling molecule is secreted by a Lux-I protein. Once the critical threshold of autoinducers is met, Lux-R proteins bind to the acyl homoserine lactone molecules. The newly formed Lux-R autoinducer complex then activates gene transcription.